Okay, to pick up where we left off, we were talking about this slide, and these are disaccharides, so two rings of what happens to be glucose, and where the glycosidic linkage takes place. So where the linkage due to the dehydration reaction, due to the taking out of water, makes a difference. Most of the stored form of glucose that we have, that humans have, is a 1,4 glycosidic linkage between the 1 and the 4 carbon. Other types of stored forms of polysaccharides, like starches, and so plant storage in the form of starch in the form of glycogen. It's not always starch, form glucose in the form of glycogen. But you also have other ways to structure this, and one of the things about this linkage is it gives itself to creating branching. And branching is very useful because then you usually have one hydrogen atom that's hanging off the end that can be broken off for immediate energy. So that breaking of a bond, think of any time you break a bond, you have immediate energy. But you also have other ways to store sugars, and that's called cellulose. The thing about cellulose is the storage, it tends to be 1,6. On the glycidic linkage is usually it's also 1,4, but it's a beta. So it's an opposite of the glucose. So you have an alpha and a beta, or opposite of the starch. I'm sorry, I'm trying to use exactly the same term, so it's an opposite of the starch. The thing about that, the alpha and the beta, so the opposite of the hydroxy group, is we have, and you experience this, we have an enzyme to break the alpha linkage, but not to break the beta linkage. So the alpha linkage with regard to the hydroxyl group, we have an enzyme to break that apart, but not the beta, which is cellulose. Henceforth, when we eat something that has that's a cellulose base, so you can think of, say, the strings in celery, or if you think of the outside of corn, we don't digest it. We don't have an enzyme that can break it apart. Other things have an enzyme that can do it, like cows have an enzyme that can break it apart, but just the difference in where the functional group is attached to the carbon, so how the carbons get together and where the functional group is attached to the carbon, can make a great difference just based on whether we can use it as a food source or not. Cellulose is still a stored form of energy, but it just hasn't happened to be one that we can access. We call it our indigestible fiber, so insoluble fiber or indigestible fiber. And then the other thing about this linkage, there's actually a third thing that can be made out of this, and this is the exoskeleton of, say, grasshoppers, certain insects, grasshoppers, spiders, things that you probably the grasshopper is the most visual, and you actually have a picture of a arthropod where it sheds its old, it grows by shedding its old exoskeleton, and it makes a very stable structural building block. This is talks about starch. I kind of got ahead of myself. It's a storage polysaccharide of plants made of entirely glucose monomers. Glycogen's the storage polysaccharide in animals. We can break down starches, don't get me wrong, but we store our polysaccharides as glycogen. Polysaccharide in cellulose is a major component in the cell wall. And one of the reasons why it's a major component in the cell wall of the plant, you actually see the molecular structure of cellulose. It's the third image down. The top image is starch, and although it's not showing it very well, you have an unbranched and then you have a somewhat branched with regard to starch. It is a slightly different design. But the middle one, glycogen, it's highly branched. As I said, that highly branching is a useful structural design because there's a tremendous amount of potential for energy. There's a tremendous amount of places where you can break bonds. And then the third one down is cellulose, and that's interesting because it's unbranched, totally unbranched. And what you can have in order to make this tough wall of the plant or anything that is structurally very tough is you can lay these strings down, these long polysaccharide strings down in parallel layers and make almost like a rope, or definitely more like a cell wall, not a rope, but just a cell wall. And you actually see an image of it. They call these microfibrils. You see an image of it, although it's very small. It's the small gray square where you have these parallel strands of cellulose, almost like weaving a fabric, and it becomes a very, very strong fabric because those links are those links between the 1,4 carbohydrates are very strong. It's the links of the hydrogens that are broken and create the energy. So, there's the term that I was talking about, the chitin, that's the structural polysaccharide found in the exoskeleton of arthropods. And think of grasshoppers with regard to that. You actually have a nice image in your book of a grasshopper. Oh, it's a dragonfly, actually. It's coming out of its exoskeleton. You have the cis and the trans arrangements. Remember, cis is active component on the same side and trans on opposite sides. We're looking at these OHs on the left for glucose, both on the, I guess, the the lower portion, and then on the trans, the lower and upper portion. And so those are alpha and beta ring structures. And then you have the starch linkage of the 1,4 starch alpha and the 1,4 starch beta. I keep getting ahead of myself. I don't realize I have all these images, but you have, remember, the alpha and the beta. You have an enzyme to break down the alpha, but not the beta, which is interesting in itself. And then cellulose molecules, strong, there's the term microfibrils, form strong building blocks for materials, for plants, the plant wall. We'll talk a lot about plant walls made out of cellulose, and you have the design there in the molecular level. Moving on to lipids. So we talked about carbohydrates, now we're going to talk about lipids. Lipids are hydrophobic molecules. So that means that they are not something that will disassociate in water. They're very important for us. They are part of our cell walls, or part of the cell walls of most living things are lipids. Because of that hydrophobic nature, they don't disassociate in water. They are very energy dense. They are about twice as much as carbohydrates. And they are useful in our body as insulation. You have a layer of fat covering your entire body, and that's a nice insulator. If you can think of it as having a big kind of down suit all over you. And then also within your body, inside and around the organs, you have cushioning of fat around organs so that they are protected from being basically from being bumped. All organs have pain receptors. All organs have the capacity to be injured simply because of force, and so you have some cushioning of organs. And lipids are very important for hormone creation. Those hormones we talked about last chapter, testosterone and estrogen, both are lipid based. It's a steroidal, they have a steroidal ring that's lipid based, as well as some of your vitamins, your vitamin A, E, D, K, your fat soluble vitamins are only taken in via fat. So fat's a very important, lipids are a very important macromolecule for life. It's the only group that's not a true polymer. It can form chains and associations, but it's not a true polymer. They are hydrophobic because they mostly consist of hydrocarbons, which form a nonpolar covalent bonds. So the hydrocarbons, strangely enough, carbon as a molecule is much larger than oxygen, but it doesn't act like oxygen. It doesn't actually have that undue pull on the hydrogen electron that oxygen has. So it's nonpolar in its covalent pull. It actually shares pretty consistently. And as I said, the most important lipids are fats, phospholipids, and steroids. Fats, kind of self-explanatory. Phospholipids, that's the structure that makes up your cell membrane. Your cell membrane is a biphospholipid membrane, and then steroids are the precursors for your sex hormones, estrogen and testosterone. The fat, we've looked at this before. There are two smaller molecules that get together to form fats. It's a glycerol and then fatty acids. So you have a glycerol and three fatty acids attached together. Glycerol itself is a 3-carbon alcohol with a hydroxyl group attached to each carbon, and fatty acid consists of a carboxyl group attached to a long carbon skeleton. And so you have have somewhat of the image here. Fats are made from two types of small molecules, your glycerol and your fatty acid. So you have on the left in the gray box is your glycerol, and what we're going to do is dehydration reaction. So we're going to take water out and make a linkage. We call this an ester linkage because of the linkage is slightly different than the one that we had before. Before, what we had was a linkage between carbons. This linkage is between carbon with oxygen in between. And what you have is then a fat molecule, a triglycerol, where you have your fatty acid, which is the orangish color, attached to what was the glycerol molecule, and now the whole molecule has a linkage, and it's called a triglycerol. So fatty acid, a carboxyl group attached to a long skeleton, long carbon chain, and there you have your long carbon chain there. That's the molecule of fat. Fats can be saturated or unsaturated. Some of this stuff probably sounds pretty familiar to you because it has kind of got into the common lexicon, and we will pick this up with the next teaching time.